Okay, our last macromolecule is nucleic acids. So nucleic acids are DNA and RNA. So a couple of, and of course, I mean, like we have chapters on genetics and DNA and protein synthesis and all of that kind of stuff later in the semester. So for now, it's just going to be an introduction to DNA and RNA. So let's talk about what the word gene actually means. A gene is a piece of DNA that codes for a particular polypeptide. A gene codes for a specific amino acid sequence of a particular polypeptide. So when we say genes contain the code for our traits, what we're really saying is each gene codes for the amino acid sequence of a specific polypeptide. So genes are pieces of DNA. DNA is a nucleic acid. Nucleic acid is the polymer, and the monomers are called nucleotides. So the building blocks of nucleic acids are called nucleotides. All right. The two types of nucleic acids are DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Protein synthesis is the function of DNA. In other words, making a specific protein or a specific polypeptide. The way that works is DNA directs the synthesis of messenger RNA, and then messenger RNA directs the synthesis of a protein. And that entire process is called gene expression. It consists of two processes, transcription and translation. So this happens in our cells. But the main thing for now is that I just want you to know that DNA functions in the synthesis of specific proteins. And the two nucleic acids are DNA and RNA. So for nucleic acids, the DNA and RNA are the polymers, and the monomers would be the nucleotides, the individual nucleotides. So this is an illustration of gene expression, and it also, I like this little chart up here at the top. DNA codes for RNA, RNA codes for protein, okay? So this first step in gene expression or protein synthesis is that the DNA molecule contains the code for a messenger RNA molecule. So you first start out with DNA, which is large, and it codes for messenger RNA, which is smaller. And that messenger RNA is small enough to fit through the pores in the nucleus and come into the cytoplasm and attach to a ribosome. So that process is called transcription. And then the messenger RNA contains the code for the specific amino acids in the polypeptide. So when the messenger RNA codes for a polypeptide, that is called translation. And that happens on a ribosome. So this is the entire process of, it's just an outline of the entire process of protein synthesis, which is the function of DNA. Now, there is a video on this if you want to watch it. It is slide 86. And again, on slide 87, the monomers of nucleic acids are nucleotides, and the components of a nucleotide are a nitrogenous base, we can just say base, a sugar, either ribose or deoxyribose, and a phosphate group. Okay, so there are two types of nitrogenous bases. There are pyrimidines, which have a single ring in their structure, a single ring, and the pyrimidines are called cytosine and thymine in DNA, and RNA would contain uracil rather than thymine. So RNA would contain cytosine and uracil. DNA would contain cytosine and thymine. So these are pyrimidines, which are 6-ring structures. Then there are 2 other nucleotides called purines, and they are double ring structures. So it says a 6-membered ring fused to a 5-membered ring. So basically 2 rings, 2-ring structures. And they are called adenine and guanine. And we basically just use letters to represent the nucleotides. So we use C for cytosine, T for thymine, U for uracil, A for adenine, and G for guanine. Okay? So these are all of the nitrogenous bases and the sugars that form nucleotides. Nucleotides, again, are the monomers of nucleic acids. So what you see for the pyrimidines is you see cytosine, thymine, and uracil, or C, T, and U. They are single ring structures, and the purines are double ring structures, A and G. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. And a single nucleotide has a phosphate, a sugar, and a base. Okay? So what happens in DNA is that DNA is a double-stranded molecule. And this is a video of the structure of DNA. It's slide 90. DNA forms a double helix shape. So it's 2 strands that are formed into a ladder, and then the ladder is twisted. I'm looking for a picture. Here we go. So the double helix or twisted ladder shape comes down to the sides of the ladder are formed from the sugars and the phosphates. And then the bases form the steps or the rungs of the ladder. And the other thing you have to know about the bases is that there is a something called complementary base pairing. And that means that A always pairs with T and C always pairs with G. So if you know the sequence of one of the strands of DNA, then you always know the sequence of the other strand because it is complementary. And you can see that here. If one strand is GG, well, that is RNA. Okay, in the DNA, if one strand is GCAC, then the complementary strand is CGTG. Okay. Video of DNA structure, slide 96. And there's some information that we're going to cover when we get to the genetics chapters, so I'm going to skip that. Okay, so we get to slide number 108. There is a summary of the key concepts from the chapter. This is a table of for the carbohydrates, and it shows the monomers of carbohydrates are monosaccharides, and our examples are glucose and fructose. The monosaccharides can combine and form disaccharides. Lactose is the disaccharide found in milk, and sucrose is the disaccharide we call table sugar. And then the polymers of carbohydrates are called polysaccharides. And our examples are cellulose in plant cell walls. Starch and glycogen are the storage form of glucose in plants and animals. And then chitin is found in the cell walls of fungi and also in the exoskeletons of insects. And one that I told you about that isn't in the notes is called peptidoglycan. Peptidoglycan is a polysaccharide that forms, it's actually got a polysaccharide component and it's got a protein peptide component. Anyway, that forms cell walls in bacteria. All right, and that's probably why it's not in the list, because it's not just a polysaccharide. All right, here we have our lipids, our chart for the lipids, and our triglycerides are fats and oils. And we talked about how they can be saturated or unsaturated. If it's unsaturated, it has at least one double bond between carbon atoms. But the structure of a triglyceride or triacylglycerol is the word that they use. I say triglyceride. But the structure is a glycerol molecule combined with 3 fatty acids. Then we have phospholipids that form the plasma membrane with a phosphate hydrophilic head and 2 fatty acid hydrophobic tails. And the way that they line up is the hydrophobic tails face each other and the hydrophilic heads face out. So they're either going to be in contact with outside of the cell or the inside of the cell. And then we have steroids, which always begin with cholesterol. Cholesterol is the precursor molecule for steroid hormones like testosterone and estrogen. So these are all examples of lipids. Then we have proteins. The monomers of proteins are amino acids. Amino acids have a central carbon bonded to a hydrogen, bonded to an amino group, bonded to a carboxyl group. And what makes each of the 20 amino acids different from one another are the R groups. Examples of proteins are enzymes, defensive proteins like antibodies, storage proteins, transport proteins like hemoglobin, and channel proteins in the plasma membrane. Hormones like insulin, receptor proteins in the plasma membrane. Motor proteins such as actin and myosin for muscle contraction, and structural proteins such as collagen. Many, many examples of proteins. Lots of examples. And then we have our, one thing, okay, so the monomers of proteins are amino acids and the polymers are called polypeptides. For nucleic acids, the monomers are called nucleotides, and technically the polymers are called polynucleotides, but you can also refer to the polymers just as nucleic acids. Our examples of nucleic acids are DNA and RNA, and each nucleotide or monomer is composed of a phosphate, a sugar, and a base, and just make sure you know the bases are C, G, A, and T. And that is the end of our Chapter 5 recordings.